recording. So by being part of the meeting, you consent to the fact that this will be recorded. We only share it with people who had an invitation to the meeting, so you can see who will all be receiving the recording. We won't share it with anyone who's not signed up for this meeting, but some people weren't able to join. So for that reason, we promise to send this around. You're also welcome to share this with direct colleagues inside of your organization, but we ask when you get the link to the video, please don't share it outside of your organization. Good. So just a sort of a basic agenda, a familiar agenda for those of you who are attending more of these info sessions. We'll do a little round of introductions, who's here, especially for our industry partners who are interested in participating in this project. We'd love to hear what triggered your interest in this project. That helps us sort of orient on why people are coming to the table and helps us as we go forward developing our content. Then I'll give an introduction to the way of working, but the real stars of the show will be Panos and Wim, who are going to walk you through the HiOver project idea. Then we'll do a little feedback round if you have questions for clarification, but even more important is sort of give us some input on how we can make this project better. And in particular, helpful for us is, what would you like to get out of the project? What would be important for you to see as a deliverable? Again, because this is a co-creation, right? Our concept of this project is very sort of HiOver and vague, and we fill it in together as we go along in the coming weeks. And then I will come back on at the very end. Hopefully we'll have 5 minutes left and do a quick summary of the next steps. And we'll talk about the timelines toward submission as we're working toward our September deadline. So with that, Panos, maybe it's actually nice to stop sharing for the introduction round so we can see faces. And I will take you in whatever semi-alphabetical order you're showing up. Oh, I have an unknown user. I'm sorry, we'll figure out who you are or is there an unknown user who identifies as unknown user? I can see you on my screen. No? Okay, well then we'll just go, whoever has not been introduced by the end, you are the unknown user, so I'll ask you to do yourself by then.
Maybe it's me.
Anna.
I don't know. Yeah. Sorry, my camera doesn't work, but yeah.
All right, yeah, go ahead. I see a name for you. Anushka, is that right?
Yeah, hi. Okay. Yeah. Sorry, my camera doesn't work, but I'm Agnieszka Nielsen. I'm from Tritomic Carlsberg Group, together with Varun here, and I'm the scientific partnership manager here. Thank you.
Great. And what triggered or were you just dragged here by Varun, or what triggered your interest with this project? No.
Sorry?
Yeah. My question, what triggered your interest for this project, or do you know what you're here for?
Yeah, I think I know I'm here thanks to Varun, actually, who was prospecting all the possible collaborations with Wageningen, and
yeah, okay.
We'll ask him why.
Yeah, we'll have to ask him why it's interesting for Tritomic. Thank you for joining us.
Thank you.
Anna Sandrini.
You hear me?
Yes. Hi.
Hi, I'm Anna. I'm working for Locker. I'm responsible for the applied research. And for the project, I will hand over to my colleague Davide, because he's the expert on proteins in our company.
Okay. Davide, do you want to go ahead?
Yes. Thank you, Anna. Can you hear me?
Yes.
So hi, everybody. I'm Davide, calling from Italy. I'm working in Loacker with Anna. I'm in charge of some sort of raw material within the research and development division, especially in the field of commodities and specialty raw material developments. We work side by side with our colleagues of product developments. And today I'm joining the call due to our interest to develop some sort of, actually to face some production issues that might be allergen management and so on. But also there is look forward to develop some sort of plant-based product that we actually have in-house. I'm in charge of proteins, so I know for sure that proteins are the focus and the target raw materials that will be employed in order to replace some sort of animal-based raw materials. Therefore, I'm here to, share our insights or understand more.
Perfect. Welcome.
Thank you very
Great to have you.
much.
Avis.
Hi, I'm Avis from CG Chaljiran, Korean CPG food company. My function is Open Innovation Manager, connected at the moment with the alternative protein team lead, also temporarily in Wageningen. We are interested in the project because not only CG is developing a variety of plant-based products, but also we are looking into proteins for clean label solutions, for example.
Great. Good to see you again. Gabriela.
Hello, everyone. My name is Gabriella. I work in One Planet Research Center here in Wageningen. And a colleague of mine heard of you, so she suggested me to come. I'm the protein researcher here at One Planet, and we are very much so interested in making part of the protein transition through the creation of new sensing solutions for proteins. That's why I'm here.
Yep. Thanks for joining. Good to see you.
Thank you.
Varun. Where are you?
Hi, everyone. My name is
There you
Varun. I work for Tritomic, so Agnieszka is my colleague. I'm a business developer here, so always looking for partnerships, both in the academic and industry space. And the main reason I'm interested in this project is because we are interested in can we use our technology to further improve plant proteins, for that matter, or any other ingredient that exists in plant protein.
Yep. Thanks for joining. Jesse.
Hi, I'm the founder of Nanomai, which is a Wageningen spin-off company. So at the moment, so we have a measurement instrument that can measure nanomotion, which we now use to detect solvents in the cleaning of oil paintings, so artworks. But it can be used for many more applications, and I think gelation or emulsion properties is one of them. So I'm looking to explore all this.
Yeah, good to see you. Really interesting new technology that we could add to the project. So the alphabetical order is slightly weird. Manon, you're next on my semi-alphabetical list.
Okay, that's fine. Good morning to everyone. My name is Manon Kooijman. I'm working as a new business manager at Kozen. And I have invited Marcel van der Vaart from our Kozen Protein division, so he can explain a little bit more why we're here, I guess.
Yeah. Marcel, do you want to break the alphabetical
Yeah, that's
order
good.
since you've been called?
Yeah, I can just jump in. So Marcel van der Vaart, I'm leading the innovation part for Kozen Protein, and that's actually a scale-up within Kozen, focusing completely on the generation of functional plant proteins. And yeah, we're always looking, of course, into leads to further improve their functionality, or maybe also look at new opportunities around that.
And a very experienced consortium partner. So if I say something wrong,
Yeah.
you jump in today. Dirk?
Yes, hi. My name is Dave van der Made. I work at Dyne Group. This is a company where we process side streams into new products, mainly side streams from the agricultural industry. And we currently have a few protein streams that we're making, which we could improve.
Good to see you here, Dirk. Getting some background noise, so if you're not speaking, please mute. Let's see. Skip past the Wageningen folks and Agnieszka. We've already had Pedro. Pedro. I don't see. Yep. Hi.
Can you hear me? Yeah,
Yes.
I come from Cargill and based in Netherlands. I'm the new ingredient and process development leader for specialized nutrition. And of course, we are definitely interested in plant-sourced proteins and other ingredients.
Great. Thanks. Welcome. Peter, also one of our experienced TKI participants. Hello.
Yeah. Good morning, everyone. Peter Hendricks from Ingredient, located out of Hamburg. And I tend to be coordinating some of the activities we do with Wageningen. And of course, we as Ingredient, specialty ingredient supplier, are also keen on this topic. And it's great to see some of our, let's say, colleagues in the room as well. For me, it would be really interesting to understand, how can we define a pre-competitive scope from which we can all benefit. So there's real collaboration in this consortium. Yeah, it's not going to be easy because the field of use is there's many of the ingredient suppliers in the room. Yeah? Of course, the people like Unilever as well, yeah, on the end applications. But to me, I mean, that's going to be, I'm very intrigued to understand how we're going to make this work. But
Yeah.
we're there to collaborate. Yeah.
Yep. Thank you, Peter. And we let's see. Maybe after today, we if everyone is on board, that's brilliant, and we will sort that out. Not the first time, by the way. Joost, my office mate, is extremely proud. He had a consortium last year with both Pepsi and Coca-Cola on it. So we thought it could never be done, but it can be done. Jan Willem.
Yeah, good morning. My name is Jan Willem Sanders. I'm based at the Unilever Foods Innovation Center in Wageningen. My background is in microbiology and biotechnology. And our interest is to apply sustainable alternative protein sources. And while we're doing that, exploring that, but we realized that we don't want to have proteins for the sake of proteins, but we want proteins for the sake of functionality. So we are very interested in understanding the functionality of alternative proteins and bringing in specific functionality rather than a protein. And so I'm happy to hear what your thoughts are and what ways you think we can move this and how we can increase knowledge in this area.
That's perfect. Thank you. You've motivated the project for me, so we don't have to do that again. Well done. Florence.
Yes, hi, I'm Florence. I'm the food application manager of Downy Ingredients, a colleague of Dirk. And I'm looking forward to learn more about functionalizing of the plant proteins. So, yeah.
Welcome.
Thanks.
Susanna.
Hi, everyone. Susanne Rasmussen from IFF, based in Denmark at our innovation center in Brabant. I'm part of the functional ingredients R&D team globally, where I'm also connected to our global protein innovation programs. I'm heading a team here focusing on functional discovery R&D activities, and among that, discovering functionalities of proteins. And of course, IFF is interested in all kind of unlocking functional potential of all kind of ingredients and also alternative proteins.
Great, welcome. Sounds very aligned with our goal. So I've had everyone but the vacuuming people and our mysterious unknown user. I'm unknown user. If you can unmute yourself.
Is it me?
Yes, it's you. Yes, thank you. Yes.
I have my name also in there, I think. What's wrong there?
Oh, I don't
sorry
know.
for this
On my
one.
screen, you're unknown user. But name yourself, and after that, we will know.
Okay, Ewout de
Hi,
Voogd.
Ewout.
I'm sorry, wasn't aware of it. Sorry for that one.
No problem.
From Algae for Food. We make healthy, sustainable, functional ingredients out of microalgae. So we're not a microalgae company, but we're more using the biomass of other suppliers to make functional ingredients out of it.
Cool, exciting. Welcome. Glad to have you, Ewout. And sorry for the name mishap.
No, sorry from my side.
And then our Wageningen All-Star team. Panos, would you like to introduce yourself first?
Sure. My name is Panos Voudouris. I'm a scientist, researcher here at WFBR. In some case, I also act as a project leader. And my focus is really on techno-functional properties on proteins.
And Wim.
My name is Wim Mulder. I've been working for almost 30 years now in Wageningen as a senior researcher, mainly in the food of protein in combination with usually with biorefinery, looking to upgrade and to valorize proteins from different side streams, from different resources, and acting mainly as project leader, and I'm involved in acquisition.
Thomas, if you could pull the slides back up. In the meantime, you all will have noticed that I am the facilitator of this process. My name is Stacy Pyatt. I'm a program manager, and as program manager, it's my job to continuously set up new collaborations in the domain of proteins and the protein transition. Good. So to the next slide, I will start to introduce the way of working in our public-private partnerships. Next slide. Yes, great. So within Wageningen, we largely have 3 types of projects, and just to say what this is not, we have science grant projects where we get most of the financing from a subsidy agency, low cash contribution, but also low influence level and no IP access for companies. Those are like Dutch science grants, the NWO or the EU ones. That's not what we're talking about right now. On the other end, we have bilateral work where we work together one-to-one with company partners. Their companies are fully in the lead. They're sort of the boss of us, tell us what to do. We do it, we hand over IP. We're not allowed to apply subsidy to that, so that is always executed at a fair market rate. What we are talking about is the mixed type of project, so public-private partnership, where we need to bring together a consortium of companies, multiple companies, together with us at Knowledge Institute. We will get potentially up to 50% financing from the subsidy agency, and there are certain ways of working that that go along with the fact that this is a public-private partnership. But first, I'd like to just go to the next slide. Talk more about these public-private partnerships. So this is about pre-competitive technology and know-how. So indeed, we have a consortium with more parts of the supply chain represented, and sometimes direct competitors together on board. And it is also a mixture. So again, it's not that very heavy science fundamental development, but it's also not fully applied sort of product development. It's really a mixture of developing new fundamental insights, but also bringing those to the level of proof of principle and model products, for example. It's open to Dutch and international partners. We are obliged to have at least one lead partner who is from a Dutch institution, and the more Dutch participation we have, the better, but it is open to international partners, especially those who do some business in the Netherlands, and I didn't hear anyone on the call who doesn't do business with the Netherlands, so I think we're all set on that. On the next slide, a little bit more about specifically what you can expect from these TKIs. So the project duration is typically between 2 and 4 years. When we want to have a PhD affiliated with a project, we are more on the 4-year side because that's how long a PhD lasts in the Netherlands. But sometimes we do a project without a PhD that can be a bit more targeted. The TKI subsidy provides us 50% of the project financing, but partners do need to contribute to participate. So the other 50% of this project we need to get from industry partner contributions, and up to 25% of that 50%, so up to half of that 50%, which is 25% of the total, sorry, needs to be in cash contributions. So together we need to come up with the financing, and then the other part of it can be in kind. Frequently asked question is, what do you mean by the in-kind contribution? We define that per partner. It can include materials, so many ingredient companies on this project. If you bring in ingredients to the project, can also include samples. If you're an end user, if you take some ingredients, you work with them, you bring them back. Sensory testing, parallel research, characterization of any samples that we make. Just also working on developments, bringing them into your company, developing new ingredients. All of that can be counted toward the in-kind contribution, and of course, the direct time that you spend engaging with the project. If this is uncomfortable for you or you want to talk about this more, please let me know. But usually we don't have a problem with this. We always come up with a plan per partner. Typically, partners each make a cash contribution of between 30 and 40K per year. But if you are an SME, a small medium enterprise, an MKB in the Netherlands, that is negotiable, and there are sort of different conditions that apply to you. So if you fall into that category, feel free to reach out. We can talk through what the options are for your participation. Depending on whether you're like really tiny or medium size, we adjust. Important for you to know, this project is not just, hey, we sit back and Wageningen does the project. We are co-creating this project and we co-govern the project. So if you are a partner in this project, we expect you to participate. In fact, you're contractually obliged to participate in the project committee, which oversees execution of the project. There we are all equals. Wageningen has 1 vote, but each of our partners also has 1 vote. And together we have the power to keep this project on track. The project committee also governs IP and dissemination, so we jointly make decisions about whether we are developing protected IP in this project or not. And also partners have approval rights over any publications that we make coming out of the project. We aim to publish the things that we're not directly commercializing or whatever is not sensitive, but whatever is potentially detrimental to your business interests, if we disseminate it, we do not disseminate from the project. And this is a super short summary of the overall IP agreement, but I want you to know this right now so we don't have unpleasant surprises at the end of the day with your legal counsels. So the principles are ownership follows inventorship, so who invented it owns it. And if we decide to patent IP inside of this consortium, and it is something that is invented by Wageningen as the knowledge partner, partners have the first right to license in their field of use. So partners all have a right to license, but in order to get that license or to ask for a transfer of a patent out of the consortium, you need to pay back at least the subsidy, and we need to document that you are paying a fair market rate for that. If this was all nonsense to you, don't worry about it. You can always approach me later. But for those for whom this is a make or break for their company, please, be aware of this upfront. Okay, next slide, I think should be then a handover to, yes, to Panos and the scientific team. If you have burning questions, if anyone has an immediate question, like, hey, some part of that really triggered me, you may raise your hand now. If not, we'll come back to a general question. I don't see anybody. We'll come back to a general question round later, or you can always contact me by email afterward. Okay, Panos.
Yes, thanks, Stacy. Well, our project is about plant-based high-functional protein ingredients. We have Stacy came out with a nice acronym, so-called FunniPro. For this project, what we think is that for the state is that plant-based proteins cannot replace the specialty animal-derived ingredients. And 2 examples that we think that is most representative is the gelatin and eggs. So we think we can do something about that, and this is by exploring new protein sources and constructing detailed structure maps, taking into account both primary and secondary processing. We will come back to that again. And we believe through this approach, we can unlock, let's say, functional potential of plant-based ingredients to deliver application-specific solutions. Now, keep this a little bit the state and the goal, and we think it is reachable because we believe in that. So this in this slide, we try to summarize in very short the reasons we believe. We need these three approach. One, it has to do with the molecular characteristics, or better saying, macromolecular characteristics of proteins. By going to the literature, we do see that plant-based proteins, there are a lot which are still unexplored. And based on their intrinsic properties, we think that they can hold the key to unlock technofunctional properties to get really functional ingredients on which can replicate the performance of animal-derived ones. The other one is we want to make use of complexation of proteins with polysaccharides. In that case, we do see in literature and also from previous studies here that by making smart choices between the physicochemical characteristics of proteins and polysaccharides, we can develop nanostructured materials on which can provide similar performance as animal-derived functional materials. The last part is about the processing routes and how to alter the intrinsic properties of functional or plant-based proteins. In that case, we do see literature and also from previous work that we have done that by playing with the processing parameters, by playing with the processing steps, and sometimes can be something very simple like we can aim for a specific pH or specific temperature conditions. We can alter the properties of what are typically known as properties of plant-based ingredients, and we can obtain more and different functionality that their interesting properties. These are the three main pillars of the project, and I'm providing here previous work that we have done and also literature that other groups and other collaborating groups has performed. This is the structure, the deliverables. The main goal of the project is to provide at the end a toolbox for achieving plant-based high functional protein ingredients. And by doing that, we also expect, we want to do that by understanding the molecular and process characteristics of the ingredients. The deliverables is functional ingredients from currently unexplored protein function or functions which are less known. The complexation between protein and polysaccharide composites or nanocomposites, and at the end exploring and applying new processing routes. And yeah, just to make it very clear, can be new processing routes, but also a combination of existing ones to develop unique functional performance. Because without saying that findings and insights from one of these direction will be used as an input for the other one. So this basically approach from the different pillars is used by combining the insights of all of them. Now going a little more deep on the different directions. So for the what we call work package 1, in this case we want to prepare or produce functional new protein fractions. And in that case, we want to go to unexplored protein sources. Here are just some examples like quinoa, oat, or oil seeds. Based on literature study and identifying the molecular characteristics of these proteins, we want to balance molecular differentiation and availability that is out. And based on that, we want to really prepare new protein fractions. This can be through standard aqueous process or can be with something more, something new. And from that, we want to do small-scale functionality testing, so screening across different new protein sources and identifying which are the most suitable ones for the applications that we need. The second part is about the complexation of proteins with polysaccharides. In that case, we want to address and prepare molecular assemblies using fibers, polysaccharides with proteins and get ideally synergistic effects. In a way, we want to get the best out of two worlds. We want to get the long fiber structure of polysaccharides and in fact placing on top of them through physical means proteins in order to gain specific functionality that we are addressing here. Have to say we cannot do everything here regarding the complexation of polysaccharides with proteins. And well, what we propose for this case is to focus really on the gelling and rheological characteristics, having in mind the molecular or macromolecular properties. The third part is on processing methods. We want to explore and apply different processing methods. For the work package 1, we refer to primary secondary processing, and here we can go a step forward. This also the secondary processing methods in order to tune the protein functional characteristics. Examples can include the temperature-induced gelation, but also going back to literature and seeing the different processing steps that have not been applied so much to plant-based proteins. Processing step like pressure has been done a lot in the past. For plant-based protein, something completely new. And in that case, we want to explore the possibilities of this such processing step. And also, use a different approach like agglomeration, try to make functional ingredients through agglomeration with this is an example with calcium ion. The first part is about physical means, right, for processing, but we also consider as a processing step the enzymatic modification, which it's up to the consortium to decide if this is something interesting or not. And of course, goes without saying that we would like also to check combination of the above processing steps that described above. Here you see 3 dots, meaning that here we list what we think that we think that are relevant. But of course, if you have more insights about that, we are totally open about that. The last part is that we want to check these high functional ingredients, to test them, to test their performance in model food applications. Here are some examples on which we think might be interesting, like baking applications, bread, confectionery, and for gelatin. Yeah, this, I don't know how they're called, but also gummy bears. A format, model format, on which though represents a real application. We want to do that for the new functional ingredients and actually compare with animal-derived proteins and see what are the knobs that we have to play in order to really replicate animal-derived proteins. That was, I believe, what we have prepared for today for the concept of this project.
Yeah, so maybe you can stop sharing again for a minute, Panos, and we'll open it up for questions or comments from the group.
Maybe a question, Marcel van Vaart, from the use of proteins from rest streams. I think there's quite a lot of activity now, of especially looking at sustainability. And if you utilize proteins from rest streams, they're often became inert because of the pre-processing that it has already seen, and they've been heated often. Any ideas or also doing something with those and maybe turning them more into functional proteins again?
Yeah, I will leave Bing to reply on that. It's more his area.
Yeah. Thank you. I'm unmuted. Yes. Yeah, indeed. Rest streams they had a history usually in temperature and processing. So usually the proteins are already denatured. So in this project we are aiming at high functionality. So we are really trying to reach that goal of this of what Panos described before, like gelatin and egg proteins. That means that we will not touch upon side streams that are had already a heavily treatment before. I don't think that will reach the goal. So we are looking more to unexplored protein sources like oat, for example.
Yeah.
Tim, could I
makes sense. Very clear. Yeah.
Tim, could I jump in to ask about, I mean, another type of side stream would be, for example, if you're doing a pH precipitation, like minor components that are coming off in a centrifugation or a pH precipitation, or I mean, my understanding was those kind of side streams, those sort of lesser known components of, for example, pulses would be of interest or would be in scope. Is that correct? I mean, not all side streams are the same, I guess is what I want to say.
No, for sure. Not all the side streams are the same, but no, pulses are in scope. Yeah.
Yeah.
Yeah, you mean the albumins then, Stacy, huh? That were not precipitated.
Yeah, exactly.
Yeah, okay.
Exactly. Yeah. Ewout.
Yeah, I have a question. Quite quick, you run into EFSA novel food limits in this part. Is there any constraint in this part for the research to be done? So you shouldn't really go into finding gelation stuff somewhere in some biomass. EFSA is quite quick and then an issue. Is that something needed for you to look at that part, or do stuff, or don't do stuff on this one?
We don't limit ourselves at this point regarding EFSA regulations.
Right. So just do, and if it's allowed, yes or no, it doesn't matter.
I would say yes.
Oh, okay.
I guess that was a bit in the point of balancing the molecular differentiation and availability. I mean,
Exactly.
Ideally, from the scientific point of view, you'd have the highest degree of molecular variation possible. So you would like some proteins that are very different looking from each other. But to also keep it practical, and usually we see that our end user partners sort of want to see, I don't know, 60 or 70% of the proteins that we're working with being things that can be available on a 2-year timeline. So I would expect from this project, that's what we see. We see a set of proteins that are available potentially available on a 2-year timeline, with some proteins that, from a scientific point of view, give us interesting molecular insights.
Can I quickly jump on that, Stacy?
Please, yeah, please.
So how do you view then the nano composites, where you actually, so yes, you have molecular variation on the protein, but then if you start combining with the other stuff, is that then per definition turning into novel food, or?
I don't think so, no.
If it's based on physical interaction, it shouldn't be. Sorry, Panos.
Okay.
Go ahead.
Okay.
Yeah. Panos, did you want to add? I'm sorry, I jumped in.
Yeah, so what we are thinking there is to use available polysaccharides, which are already available for food applications with EFSA approval. Then we want to play with physical knobs. This can be electrostatic interactions, can be hydrophilicity and hydrophobicity. But we want to play with the physical interactions in order to make composites or maybe the term nanocomposite is scary for this, but we want to make a complexation in a macromolecular level, and this does not necessarily mean a new ingredient. It's a combination of 2 existing or more ingredients. Yeah.
David, you had very politely had your hand up. Sorry, we.
Yeah, thank you. First of all, thanks for the presentation. Maybe I can speak from a final user eye. For us, it's very compelling to frame the macromolecular structure of any kind of botanical source of protein and see the comparison. We, as a LOACer, we have long history of using egg white for one kind of our products. We are looking for specific foaming properties, so not actually gelling properties. We know there are 2 kind of egg whites in the market. One has foaming properties and one has gelling properties, according to the pre-processing they undergoes. However, we're looking for the foaming properties, so not the whole egg, but just the egg white. I'm not really following the combination of ingredients because that might work. for instance, combining proteins with polysaccharides. We have a specific direction into not using extra additives for our products. So our main focus is to find out whether there are alternative from a plant-based proteins that might replace egg white with just one ingredient. I know that it's a very complicated and challenging activity, but we are quite into it, and we look forward to find a solution. For us also, moreover, there are 3 other properties that we're looking for. This protein is better not to be an allergen, because otherwise we replace an allergen with another allergen. This protein is better not to increase the density and the viscosity of the dough, of the paste. Otherwise we have issues, but that might be a topic for further developments. And the final target properties we're looking for is that this protein, this foam that has to be developed, it's better to be stable during the baking process. And that's another challenging topic we are facing now.
So before I let Panos and Dim respond on content, I'm going to use this opportunity to say one of the things we do as follow-up after today is find out who are those partners with the closest fit to the content who are really motivated for this topic, and do some deep dives where we really dig into the content. We take an hour, we dig into the content, we go through all of the boundary conditions like allergenicity, et cetera. We discuss how to handle them. So I'm already mentally volunteering you, David.
Yeah, I just wanted to give you a quick overview on what we are looking for. Actually.
Yeah. So then, Panos, maybe you want to reply on the content a bit.
Sure. Well, by the way, we are looking for such boundary conditions as thanks much, David, for that. Polysaccharides, I think is mostly for the gelatin part, not necessarily for the egg white. Wim will know better about that on what are the most promising protein source for replacing egg white specifically. And now for the foaming properties that you mentioned, I think their proteins as alone will have much more chances than these complex carbohydrates, complexation with the polysaccharides. Yeah.
Fair enough. Thank you very much.
But we can, as Stacey mentioned, we can go to more in-depth discussion during the follow-up meeting. Yes.
Perfect.
Yeah, to add on that, I mean the setup that we choose is to explore the possibilities of unexplored protein sources, and we will do that in a way that we choose a quite straight at the beginning of the project. At least that is what we envision, a quite straightforward extraction process. More or less in line with Osborne fractionation, just simple, but in that way it is quite standard. And when there we can make quite some different samples from the different biomass, different protein sources, let's say. And then we will produce these fractions, and we will add that into our methodology how to characterize the functionality. And foaming is standard one of the characteristics that we study. We don't know it up front. That is also why we would like to explore this in a pre-competitive way. But yeah, I mean there are already also some. For example, oat drinks on the market that are being used as an alternative for foaming in cappuccino and this type of thing. So I think we can learn from those type of technology.
Excellent. Oh, Ewout, I'm sorry. I thought that was a legacy hand. You have your hand up again. Yeah.
Yeah,
jump
please.
in.
Yeah,
Yeah,
I was
sorry.
it didn't work out very well. Oh, yeah, I'm still open. I'm a little bit searching. We have some extractions out of the microalgae which show amazing gelling properties. I definitely want to know more about it. Okay, what is it exactly what's going on there? But also in a practical approach, you really want to bring it to market as soon as possible. I don't have 4 years to get answers on this one. So I'm really, it sounds like, oh yeah, it's something special functional is going on. You really can research it, really can think about why. But I'm looking for the match in this one. So I feel that there is a lot of answers can come from this project, also to understand what we have. And I definitely need also that knowledge to explain the clients later on. But it also feels like a mismatch on speed and need. But please, how can we make this happen? How can we make this work?
Yeah, thank you. Thank you for raising it because it is something that we navigate together inside of the TKI in the public-private partnerships. It is not uncommon for us to have short-term target, like first-year deliverables based on the current generation of ingredients from our ingredient suppliers. So if we don't need to go through a process development, if we don't need to go through preparing fractions, that application work package in the first year is actually often very oriented to characterizing what can we do with what we have right now, while the applications work package is waiting for new things to come in from the other more sort of process focused or more future forward work packages. So it's not uncommon, and we will have both short-term and long-term deliverables defined for the project. And what I hear from you is, hey, it's really important for us to have some short-term deliverables to understand and verify what our current extracts and ingredients derived from our algae can do, ASAP. That's entirely possible to build into the first year work package.
It's also in your interest, what I understand, is in the interest of this project.
It's a tip. I mean, it's a very, yeah, look, I don't know, Wageningen, our interest is to run a really great project, right? So it's a collective interest, like it's a co-created project. Yeah. So I think, well, Wageningen is one, maybe if we have 8 partners, Wageningen is 1/9 of the interest in the project, right? But of course, it is, the TKI subsidy is meant for fundamental and applied. So it will be a combination, right? We want to test where you are with your generation right now, generation 1 ingredients, but we also want to develop some knowledge that's going to help the ingredient partners to develop next generation ingredients that are even better. Yeah, thank you for the question. I think that's also a typical an FAQ, so I'm sure it was in someone else's head as well. Jan Willem, you also have your hand up. You're muted though. I see you talking.
My question is partially on the same line. I understand that you want to study crude proteins from plant sources, and also moving to partially purified fractions and extracts. We also consider to make the next steps and to look at the functionality of single proteins for the sake of understanding and for the sake of having even better functionality.
Yeah, what a perfect follow-up question, because this is the line we're walking between short-term deliverables and long-term knowledge development panels. I guess you are made happy by this question, no?
Yes. By single proteins, you're referring to proteins coming from a microbial biomass, right?
Oh, yeah, I understood it differently. I understood it also differently. Did you mean also like instead of a pea protein isolate, would we go to visilin, convisilin, what are they? To the individual, also that.
I mean this pure single protein, yes.
Pure single protein, yeah.
We are more than happy, yes, for that. Also, yeah, with Vim, right, for a specific biomass, we can extract really the individual proteins from there, and figure out the technofunctional properties of really single proteins. Yeah.
Yeah, that is one of the things that we were. I mean, just as an example to mention, we are running at this moment another project on extracting proteins from oil seeds, And we have, for example, we have we are able to produce a protein fraction with a different isoelectric point. Yeah, because most of the proteins, they have an isoelectric point around 5, let's say, and this one has specifically around 7, 8. So in combination with carbohydrates, that could be a very good property to increase the complexity because the charges are more compatible with each other. So yeah, we're looking into for sure into those type of fractions, call it single proteins. Yeah, I mean, what is in the end the definition of a single protein? But indeed, I understand your question. Yeah, yeah.
Yeah, and in many cases, I think we need to get to that level if we want to have this very specific targeted functionality.
Exactly.
Yeah.
Yeah.
Yeah,
Well,
that's
to
a good
actually
one.
understand which fraction of the ingredient is doing the job.
Yep.
Exactly. Yes.
Yeah. Good point. Totally agree.
And my other question is on functionality. I hear you talking about gelation. Is that the one functionality that you want to work on, or are you open to explore other functions?
Yeah. We are doing the almost small scale benchmarking, meaning that we are focusing on the main first technofunctional properties. This typically is solubility, gelation, emulsification, and what is it? Solubility?
And foaming.
Foaming, yeah, exactly. These are the main ones, and then if we do see a good performance, then we go much more deep into this.
Yeah, and companies can also help us in this, because David, as you mentioned before, to have stable foam during the baking process, that would be a very nice thing also to. I'm already thinking of how to do in-kind contributions, but that would be a perfect thing to determine.
Yeah, from a practical way, we can definitely help.
Yeah. Yeah, and I think that also goes to the concept of co-creation, right? Should it happen that everyone says, well, actually gelling is not relevant for us, and then
we drop.
Yeah, we work to sort of focus on what has the priority also for our partners. So please remember Wageningen is sort of one voice in this whole collective of designing the consortium. Susanne, I love that because we haven't heard from you yet. So please jump in. I will make this the last question, though. I'm so sorry for everyone else who has great questions, I'm sure, but in light of time, please.
Yeah, it was just a comment. So for example, the application, right, it's a complex system, right? So we know that pure functionality when we test it in the labs is one thing, but then when you add it into a complex matrix of different ingredients with different salt levels and whatever, right, then it becomes a bigger challenge to keep that functionality stable over a long time, right? So stability over a long time in a matrix is important, and then it comes back to the scale we're aiming at when we want to look at these processes and have potential protein fractions available to actually look into this. So what are we aiming at here? Is it still bench scale, or are we looking at also going to bigger scales?
So we do have some. Oh, yeah, sorry. I will just say because it's a TKI, a subsidized consortium, we typically do in Wageningen end at the model product, and we talk with the industry partners about what their in-kind will be. But when we have end users, we love to see those benchmark products. And Wim, you can, of course, with your many years experience, can talk about that further.
Yeah, well, of course. I mean, I think at the beginning it's good to have to have a number of samples, and then in the after some screening, then some of those fractions they seem to have good potential. And then we can scale it up. I mean, that is what we usually do in projects. And yeah, let's say 1 kilogram of scale can be possible. But of course, it's all also on time limitation. Where do you put your your focus and your effort of the whole research team on. And that is, I think, also a combined decision. But this thing with the complex matrix situation, I discussed it also with Panos, and we are not only looking in the pure nicest way of the environment, we also checking the pH and the things like that too. We will not go to end products because that would be more like a bilateral project, I would suppose. But we are for sure also taking the matrix into account. Yeah.
So Panos, could you pull up the timelines? Thank you for that complete answer, Wim. 1 minute left. I will run through the timelines of what's going to happen next. So we are now heading into June. And for the next 6 weeks, what we're going to be doing is some deep dives, defining the project content a little bit more. I will ask you I will reach out to you and ask you to respond to me with an answer, yes or no, whether you want to be part of co-creating this project. Ideally, we'd like to have at least 4 enthusiastic partners who want to work with us on co-creating the project. If we don't have anybody who's enthusiastic enough to invest their time, that's also a sign to us that maybe you thought you were here out of curiosity, but not committed to co-creating it. So please, if you think this is a nice topic for you, put up your hands and join us for that. Also, just an open invitation. If you conclude after today that this is not the project for you for some reason, please tell us as soon as possible, because it's far better for us to have an accurate head count than it is for us to sort of troop on towards September without without genuine commitment. So no's are welcome, yeses are very, very welcome. And we love to work together with you on co-creating it. Toward the end of July, we'll have to do a go, no go heading over summer. So we'll have a sort of head count, at least by then, on who's committed to this project. Our deadline is September 1st, which is horrible for the European summer holiday season, which is why we need that good head count in July so that we can sort of run through the last part of it over summer while people are away. So we need your firm commitment ideally as soon as possible. We get subsidy grant notification about 6 weeks after the deadline. So as we're heading through October, and then we head into the contracting process, October typically to March, and ideally kick off end of Q1 2025. Most important is I'm going to send you this recording. I will send you the slides, and I will ask you in that, please respond to me if you're willing to work with us on this concept proposal further, and or a no, also very welcome. I love respond emails, so the only thing I ask you not to do is just ignore this message from me. If it's a maybe, send me a maybe, that's fine too. But just to get a head count on where you are would be much appreciated. That's all for today. I'm sorry we didn't have time to hear from everyone. If you had a question that we didn't get to, also send that to us. Panos and Wim are always happy to hop on a short call and clarify things or dig into it a little bit more. That's it. Thanks so much. Always 2 minutes overtime. That tends to be the habit here. Thank you for these extra 2 minutes, and we look forward to being in touch in the coming weeks.
Thank you very much.
Bye-bye, everybody.
Thanks for the intro.
Bye-bye.
Bye.
Bye-bye.
Welcome.
Bye-bye.
Bye-bye.
Bye-bye.